Chris? Okay.
Welcome to the HNRCA Monday Seminar Series for March 3rd, 2025. Before we begin, I want to remind everyone to leave their questions in the Q&A tab. Today is my distinct pleasure to introduce Tufts' very own Dr. Sasha Poltorak. Dr. Poltorak is professor and chair of the Immunology Department at the Tufts School of Medicine. Among his many contributions to science, as a postdoc, Dr. Poltorak used classical genetics to identify toll-like receptor 4, better known as TLR4, as the major receptor for bacterial lipopolysaccharides, an enormous discovery that has shaped much of modern immunology and resulted in the awarding of the 2011 Nobel Prize to his mentor, Bruce Beutler. Following this landmark paper, we were fortunate enough to recruit Sasha to Tufts in 2003, and he has since authored key contributions to the areas of inflammation, cell death, and the genetics of immune responses to pathogens. Today, we will hear more about his work on the genetics of immunology and immunity with his talk entitled Battle of the Sexes, Mendelian Genetics of Sex-Specific Immune Responses. With that, Sasha, the floor is yours.
Thank you very much, Chris, for such a nice introduction. And as Chris mentioned, in my lab, we enjoy and we take advantage of constant monitoring of cells and cells living and cells dying. And you can see this image on the right. It's the most advanced technology, phase contrast, which can provide really insight into the cellular compartment and in a very non-invasive way can tell a lot about the cell fate. And as geneticist, I'm using wild-derived mice, which I will be telling more about in the next few slides. So the sex dimorphism in immune response is well documented and well established, and males have much more profound and robust innate immunity, whereas females' adaptive immunity takes advantage of the high CD4 positive T cell count, more interferon gamma production, which synergizes with innate immune functions. On the other hand, the trade-off is autoimmunity and autoimmune diseases, some of them attributed to the genes residing on sex chromosomes. While we age, these hormones become major players and definitely change a lot of adaptive and innate immune features. And for example, testosterone is known to predispose to cancer and to infectious diseases and sepsis. So it's well documented that men spend more hours, more time in intensive care unit than women. On the other hand, as I said, the trade-off is much more high prevalence of autoimmune diseases in women and more interferon production mediated by innate immune genes, mainly TLR7 and TLR8, that are encoded on X chromosomes. And so in this case, lionization or silencing is not like happening 100% in all women. So the trade-off of immune response, and in this case, I put LPS response just as maybe the most used, the most famous, but at the same time the most notorious model because of discrepancy between human and mice responses to LPS and susceptibility to infection. And the common regulators of this sort of balance are immune genes, and the role of these genes in reproduction is getting more and more established these days. In this slide on the right, the researchers infected. They gave sublethal dose of H1N1 influenza to mice, and they showed that in fact mice, uninfected mice, they had healthy green bar, healthy progeny, and well nutritioned, and at the same time, the picture on infected mothers delivering offspring changed dramatically, and this 50% of the dead offspring was born, and 25% under proper age. So only 1/4 essentially is born with a normal weight. So the evidence in women is well documented, and there are reports that on average mortality among pregnant women is 5 times higher than in non-pregnant women from the flu, and there are published reports about that. The activity of NIH Women's Health Office is fantastic, and this office is charged with the idea that in fact to change bias which was for a long time present in scientific community, to use males as main control, and to sort of shift this balance towards investigation of females and particularly women health. So what are the genetic components of this sex dimorphism which I mentioned? And are there any autosomal factors? Because most of the discoveries happen in the context that, the traits are different, but the traits are conferred by genes. So where are these genes? And are there common regulators of reproduction and immune response? So I want to take back to very well-known geneticist Theo Dobzhansky. He's a Ukrainian-born, emigrated in U.S. and spent very productive time in North Carolina. And the famous saying that attributed to him was, "Nothing in biology makes sense except in the light of evolution." So what we can take from that is that whatever traits we are working on, whatever we are liking and definitely prefer to study, the survival is the main challenge in the context of evolution, and that's perhaps the only factor that the nature cares about. And in these days, there are too many traits to be monogenic. So conceptually, we should be moving very fast to the idea that many more traits are multigenic traits, and we need a model to study. And the most appropriate model, of course, is human model, but humans are separated by just 200,000 years. So in terms of evolutionary distance, that's not very dramatic difference. And so how does evolution fit into the survival, and how fast is evolution? So in this slide, I showed you the agouti gene, which is sort of evolving in mice, mostly on the Florida Peninsula, and they are dependent on the environment, whether mice reside on the beach or whether they are in the green outfield scenery. They change their color, and so this happens within several tens, thousands years. So it's a very quick process. So evolution, if it's challenged by the fitness, it's working very fast. I'm personally, and perhaps many people, many researchers are concerned about innate immunity and how it is evolved from across the species. So again, the question is, are these genes which work similarly in invertebrates and high vertebrates, do they evolve or they acquire different function? And here, as Chris mentioned, the very same gene, a toll gene, and this is Kristen Nussland-Wolgert. He exclaimed toll when she saw this mutant in toll. So it confers dorsal ventral development in embryo, and in mammals, it's a product of LPS gene, TLR4, one of the members of toll-like receptors family. So and apparently had nothing to do with the development, but mostly with the sensing of gram-negative bacteria. So does this gene preserve its function, or it expanded its function, and how does evolution fit into the changes in function of this gene? So to address all of these questions, my lab takes on so-called wild-derived strains, and this is a mouse of mole strain is pictured. They are different subspecies of mice, and they are so different that polymorphism happens every 100 base pairs. So essentially, you can say that 1% of the genome of these mice is different from 57 black 6 mice. And they're different not only in weight, they're different in responses and in innate immune responses. They are much more close. I won't just remind you this in this slide that mice in general, they're more close to humans than cats and dogs and pigs and rats. So essentially, in terms of the evolutionary distance, mice are very appropriate model, perhaps, in terms of diversity. And that's the first data slide where I want to show you that we challenged B6 and mole parental strains with LPS, with a lethal dose of LPS, 25 micrograms per gram of body weight, and we observed both of the group of mice succumbing to the LPS-induced lethality. And F1 mice produced from the breeding of these B6 and mole parents, they were absolutely resistant, then couldn't care less about the dosage. I'm not showing you the data, but we couldn't find the dose that kills these mice. And that was accompanied with the temperature. So hypothermia is a hallmark of LPS response in mice. And unlike humans who increase their temperature, the body temperature in mice decreases. And within a span of 7 hours, we can see that in both parents, B6 and MOLF, essentially the temperature decreased to the level that for the purpose of humanity, the mice were sacked. And F1 mice, if anything, there was a little bit of increase. There was a little bit of increase of temperature also in MOLF parent, which are, as I said, genetically diverse from B6. And that tell us that told us that perhaps these mice, MOLF mice, they find a way to die, but these ways is perhaps different from the way B6 parents were dying. And then we did something very common, what we do in the lab. We produced a lot of F1 parents and we bred them. And that's where at the level of F2, we generated large panel of F2 intercross mice, and the genome in these individual F2 mice was different, so that's where the shuffling of genome happens. And then we challenge them with LPS. So that's the picture of 106 mice in the first panel of F2 that's showing the diversity and the breadth of the temperature decrease. And again, some of the F2 parents, similar to MOLF, they recapitulated this increase. We applied a linkage analysis to this F2 panel to identify several loci in the mouse genome, and these are the numbers of chromosome, but with the highest linkage to the locus on chromosome 17, which encompasses two genes encoding hormonal receptors, so FSHR and LHCGR. So these receptors are encoding receptors to follicle stimulating hormone and luteinizing hormones. Both hormones are very important in sex development in both sexes, as well as in follicle maturation. And they are known to promote, because of that, in the course of that, they are known to promote production of estrogen. So the hybrid vigor, which I characterized first, it's a used to be very relatively popular term, but well forgotten, and certainly not very popular in mouse genetics. But essentially, many plant geneticists use these when they introduce new features in hybrid plants not seen in the parents. So this resistance in F1 was, according to this model, was encoded by two loci. So we were very surprised that actually the mice heterozygous for the locus, for this chromosome 17 locus, it was sufficient, a single locus to confer resistance to LPS. As you see, this is a temperature, and the mean temperature in the group of mice heterozygous for this locus is higher than in both homozygous groups. So these data were telling us that actually one locus, as compared to two loci, one locus was sufficient to recapitulate this phenotype of hybrid vigor. We then separated F1 females and F1 males. So we put them separate from each other, and we looked at them at their, again, mean temperature, and we were almost shocked to see that F1 females, and so don't forget, these are genetically identical across all the genome group of mice, right? F1 males and F1 females, and F1 males behave as they're supposed to do. Their responses, their temperatures, endpoint temperatures is very tight, whereas there is a certain spread in females. And at the first time we accounted these to perhaps different level of estrogen in females, but really role of estrogen, I mean, to that extent, to show such difference was not documented in the literature, and then we took F2 females and F2 males, and we saw that actually, again, the mean and the difference and the more predisposal of F2 females essentially to LPS susceptibility is significant. We then separated females and males. So in this graph, there are three groups of females and three groups of males, and we separated them by the haplotype at this hormonal receptor locus, and we saw that homozygous mice are very much different from the heterozygous females for this locus, statistically significant. Again, the takeaway message from this graph is that essentially this locus was specifically conferring LPS responses in females, because in males there was no statistical significance, and I could say that males couldn't care less about the haplotype for this locus. So we then used so-called CAPE analysis. So CAPE analysis is an advanced mapping approach which my collaborators at Jackson Lab, Anna Tyler and Craig Carter, they developed. And what CAPE does, it introduces integrated readout instead of individual readouts, which could be temperature or different cytokines. So they found a way to put all of these readouts together, what they called eigentrait. And there could be many different eigentraits generated based on different contribution of these individual readouts. So using this approach, they found many interactions in the genome, and particularly with chromosome 17, I want to to bring your attention to chromosome 2 loci, and one locus contained in the peak FSHB. So that's a beta unit of FSH. It's a protein, so it's a protein hormone, so it's encoded by a gene. And then another gene which was on the distal end of the chromosome 2 was ZBP1, a very well-established sensor of Z nucleic acids. So Z nucleic acids are the ones that are improperly swirled, and actually they are swirled in the other direction, and they're detrimental, and as a sign of alarm, they have to be blocked and neutralized, and ZBP1 is one of the sensors that is involved in that. And what about males? So quickly about them, I showed you the data that they responses in males didn't map to chromosome 17, but they map to chromosome 12, and encompassing the locus with the paternal inheritance. So that's an imprinted locus. It's one of the largest loci in the mouse genome, which has several genes paternally inherited, but also the other group of genes maternally inherited. And so the trait in males was inherited actually in dominant manner for moles. So it had nothing to do with the hybrid vigor which we observed in females. And the most gene of interest for us was DLK1, which I can mention later if we have time. Hmm. I did something and it stopped by some reason. Okay. Okay. Well, to make a long story short, we identified skipping of 9 exon in LHCGR in one of the genes encoding hormonal receptors, and this skipping was detected in RNA sequencing, and these are sashimi plots. So sashimi, they look like sashimi because these connectors they link together different exons if they happen to be in the same read in RNA sequencing. And you can see that MOLF is a donor of this skipping. It has minor skipping, and B6 doesn't have skipping of exon 9. This is exon 9 at all. But this skipping amplified in the actually much more robust in F1. So I want to remind you now that this, in fact, molecular behavior of the RNA splicing fits into the hybrid vigor model where hybrids have more profound feature not seen in parents, and this has been confirmed by qPCR. So what does skipping of exon 9 do in this receptor? There is a hinge region which is LHCGR is a G protein coupled receptor, and it has 7 domains, and it has many different activities in the C terminus of the intracellular portion of this receptor. But it also has many leucine-rich domains which are separated from the surface by hinge. And so exon 9 deletion takes care of this hinge, removes the hinge, and the predicting modeling shows, well, at least makes us believe that there is a constitutive. It makes receptor constitutively active because then this extracellular portion of the receptor is more positioned more close to the surface. We cloned and activated 293 cells with this full-length copy of receptor and with the truncated isoform, and we showed that the signaling, the CIMP production downstream of receptor is essentially is always at the stable without even activation of the receptor is present in this truncated skip9 exon version. So it looked to us that this alternative splicing really results in the constitutively active form of LHCGR. In the first slide, or in one of the first slides about the data, I showed you that levels of cytokines in the circulation of F1 LPS-resistant mice were low. And so the question was whether their cells are refractory to macrophages, mainly to LPS insult, or there is less macrophages in the circulation. And this panel shows that, in fact, it was the latter. So in here, we went to the bone marrow, and using multicolor flow cytometry, we analyzed most of the CD45 positive cells. So CD45 positive cells, it's a common marker for leukocytes, including lymphoid and myeloid cells. And in the bone marrow, I was not aware of that, but it was certainly what's well established in the literature. The most abundant cells are B cells and neutrophils, and neutrophils essentially they develop in the bone marrow, and then they come back to recycle, to die, and the speed of this replenishing is astonishing. So you can see from this plot that F1 gray bar is significantly lower for B cells and for neutrophils and for inflammatory monocytes in the bone marrow. But when at that time we acquired from the collaborator LHCGR knockout mice, so F1 mice, they had constitutively active form of LHCGR. LHCGR deficient mice, they have no receptor like expressed. And you can see that these knockout mice, they have even high, if anything, they have high numbers, high incidence of CD45 positive cells in the bone marrow. So it looked like this receptor, in addition to the gonadal functions of this receptor, it was also involved in regulation of the resident cells and perhaps in homeostasis. We then went to the spleen, another immune tissue, and we took CD11b positive myeloid specific cells, and we showed that, in fact, these F2 mice, so as compared to F1 mice, we went to the F2 genetically diverse mice, and here we showed using not large enough numbers, but enough to show statistical significance that, in fact, the numbers of these splenocytes, myeloid cells in the spleen, are lower in F2s that are heterozygous for the receptor, but in both in the littermates which are homozygous for B6 or for MOLF, these numbers were higher. So again, we not only were trying to explain the phenotype in parents, but it looked like this behavior fit our mapping data. So LHCGR receptor was in fact involved in regulation of the incidence of the myeloid cells in the spleen. And then we identified expression of a receptor LHCGR on the surface of B cells. And again, mice with the heterozygosity for this receptor, they had high expression of this receptor. In concordance with the resistance of mice with constitutively active LHCGR, mice that are deficient for LHCGR, they were actually hypersensitive to LPS. So the idea would be concordant with this plot, which shows high numbers of perhaps myeloid cells in the circulation of these mice. And that would be my explanation that of heightened susceptibility in these mice. So the model we have now, and it's, as they say, in progress, is that pituitary gland, which is the main source of luteinizing hormone and follicle stimulate hormones, either directly via receptor expressed on progenitors, bone marrow progenitors, or indirectly via stromal cells, promotes decline of lineage-specific fully differentiated CD45 positive cells in circulation. And it may get some contribution from estrogen, which in general is known to contribute to make responses in macrophages refractory, but it was never seriously studied in a sort of genetic basis, and essentially no one looked at a long-term exposure to heightened level of estrogen. So our model would be that F1 females because of constitutively active LHCGR, they have high levels of estrogen, which is conferred by their high fertility. And then estrogen can be also involved in hematopoiesis in the bone marrow. Now, to get back to the single locus versus two loci, the conundrum perhaps to be solved and to explain how one locus was able to sort of recapitulate the phenotype seen conferred usually by two loci, we have to go back to the genome of MOLF, and these MOLF mice were product of a so-called introgression. So introgression is as compared to natural hybridization, which is a component of evolutionary development. So introgression is a manual mixing of two different genomes. And in case of MOLF, two genomes, mus musculus musculus and mus musculus castaneus, as you can see, they contribute mostly to a MOLF genome, with the domesticus subspecies and C57 black 6 are representative of these classical inbred mice. So contributing very little. And you can see relatively large patches on chromosome 17, patches of castaneus genome. And in fact, chromosome 17 LHCGR locus is represented by castaneus. So these patches are signs of introgression, and so it imposes some challenges in fertility, and we hypothesize that perhaps some de novo mutations arose in MOLF genome to account for this splicing. And we have now, again, this is something in progress. But you can see these at least four identified SNPs different from castaneus, and they are close to the donor splicing site in the exon 9. You can see exon 9 could be skipped because of these SNPs. So in support of this hypothesis, we did genome-wide incidence of polymorphism in MOLF, and LHCGR locus went into the 92 percentile, so it was in top 10% of genes, highly polymorphic. So like the model would be that because these mice were introgressed in the lab, and they were developed in the lab in Japan, in fact, they had to face some reproductive and fertility challenges, and some loci, and particularly these locus, which encodes hormonal receptors, had to undergo significantly heightened mutagenesis. I mentioned and promised you to get back to ZBP1, which I find particularly exciting as interacting with LHCGR. And the example of this contribution is that, again, this is LPS resistance in terms of temperature, and that's the lower, the lower resistant. These mice at the level of -1 are highly sensitive to LPS. So you can see that MOLF homozygosity for ZBP1 almost kills mice with the LHCGR homozygosity, but heterozygosity for ZBP1 rescues. And so when we then arranged all heterozygous for ZBP1 F1, F2 females, we found another gene which is called MOF10, almost completely and statistically significantly associated with the haplotype. So two haplotypes in F2 mice for these genes, they almost matched each other. We looked what MOF10 is, and it's a helicase which can be which is involved and responsible for unwinding the nucleic acids, and we hypothesize that it can be involved in maintenance and homeostasis of Z nucleic acids. So we silenced MOF10 in fibroblasts, and we found a much more increased staining for the Z nucleic acids. So we believe that ZBP1, using ZBP1 as a BITE, we identified MOF10 as a novel regulator of the Z nucleic acid. And maybe I sort of ended a little bit earlier, but I want to like maybe go over the main finding of the work, that we identified alternatively spliced isoform of luteinizing hormone receptors, and these given biogenesis of luteinizing hormone receptors, and as well as follicle stimulating hormone receptor, skipping exon 9 potentially can explain phenotype. Splicing correlates with aging. So I didn't show you the data, but in fact, aged females, they become sensitive to LPS, and that matches perhaps decrease in hormonal levels in females. And in mice, these decline and gaining back responsiveness to LPS starts at age of 1 year. The problem with the field is that estrogen is notorious to detect, and it's in mice, and there is a very little reagent base for these, and so we are trying to set up assays with the mass spectrometry to be in control of the estrogen production in these mice. And so FSH and LH1 of rare autosomal sex dimorphism regulators of the innate immune response. So as I mentioned to you, locus on chromosome 12 confers LPS responses in males. So that brings really interesting situation where you have two different autosomal genes conferring responses in sexes in sex-specific manner. And perhaps given the number of gene-gene interactions that we obtain for the females, and I didn't mention, but we accounted 23 additional genes which make this bimodal distribution of ZBP1. So I think this inquiry could be further extended, and so potential splicing in humans, there is such, and actually exon 9 has been reported to splice in human individual in male, but it has profound consequences in sex development, but nobody looked, of course, in the innate immune responses. And given the extent to which patients are treated either based on fertility issues or on birth control issues, I think this work is alerting us to look at the consequences, because if the doctor puts a patient on a treatment for or against fertility, like the trade-off certainly has to be taken into account, particularly if these hormones have really very high affinity, and they do. Affinity KD for both hormones is in a single nanomole, so they are pretty potent activators of these receptors. This work wouldn't be possible without two talented researchers, and that's a postdoc, Hikaru Tamura. He came from Japan for 3 years, and we are trying to finish a paper now. And Haley Mendelian, unlike Hikaru, she stays with me now for almost 10 years, and she came to the lab in 2015, and she evolved to the level of research assistant professor, and now is trying to apply for her own funding. And this work has been supported by 2 grants from the NIH that I had. So thank you very much for your attention.
Thank you so much, Sasa, for such an interesting and stimulating talk. While we wait for the Q&A, I'll take the host prerogative and ask you a couple of questions. In particular, I want to draw you back to your discussion of DLK1. This is a gene that I was very interested in graduate school because I studied it as a genomically imprinted gene. And I'm very curious about whether you saw imprinting showing up as a general parent of origin effect type analysis in your study here.
Yes, absolutely. I mean, this is fantastic. And, once I saw, I was, I mean, I was not in disbelief, but it's one one thing to read about imprinting, and like in this case, it's paternal inheritance. And then we knew that our father was male, was MOLF, so we saw 95% of in the reads were MOLF specific reads. Yes.
Did you end up seeing any other imprinted genes in the process?
We didn't go further because, in fact, I'm trying to renew this grant, and I was discouraged, well, conceptually and mentally, that made me not think about the male-specific genes. But I found this locus very, very exciting. Yes, yes, I would be happy. Like the purpose, I thought to arrange my talk so that I could perhaps infect some people with these genes, and obviously, um, mm, it's very robust readout, particularly with this CAPE analysis, because it brings a lot of observations, and so we can talk about this, and I'll be happy to share all information that we have about chromosome 12.
Wonderful. So Daniel Promislo asks, first he states, fascinating work. To what extent might these effects that you are discussing influence the strong male bias in mortality that we see throughout the human lifespan?
Well, perhaps it needs to be, yeah, it needs to be seen in the context of aging, and I didn't age these F2 mice, because F2 mice are the ones which are really sort of the meat of this grant, because using F2 mice, I'm able to say something about genes, right? And separate F2 mice into sexes, into males and females. So unfortunately, we didn't do a lot. Uh, Again, the power of F2 approach is that you assort F2 males or females by certain loci, but then all other loci become playing role. And that's amazing because like assortment for a single locus could sort of reveal contribution from other loci. And I think phenotypically, F2 mice are very different. So, I mean, I'm hopeful that, to answer the question, I'm hopeful that detailed studies of this feature in mice would be able to address some of the questions and some of the observations that that were made in human population.
Wonderful. Well, I'm going to give everyone a moment to answer or put out any more questions and take another opportunity to ask more questions along my own interests. So at the you talked about the relationship between fertility and immune responses, and you of course showed the elegant graph of the relationship between development and influenza infection as a classic example of this. Uh, do you think that the major effect of LHGCR is in the relationship between the survival of the female versus the development of the offspring? Or is it more about the, maybe I'll just stop there and say, do you think that's the major battle there is between the offspring, or do you think there's a male parental contribution to it?
Yes. Well, thank you for this question, Chris. It's we keep thinking about that and discussing, because in pregnancy, certainly all brakes are off, right? All hormones are through the roof, and perhaps again, we thought, and sort of the field uh, is concerned about the role of hormones in a short timeline. Like, many papers that report contribution of hormones, they end up adding estrogen, for example, to the macrophage culture, and like observing some things. But this is homeostasis, so it's a much more long-term process, which takes perhaps days, if not weeks, to establish. So these F1 mice and F2 mice and pregnant women, they stay for weeks, right, on high level of hormones. So they have to be perhaps different incidents of bone marrow and resulting in different numbers of the resident macrophages in pregnant women. That we see now, and we start asking questions whether phagocytic activity of neutrophils, for example, is different, because it's quantity versus quality. So under constant pressure of hormones, we really don't know yet how much, to what extent quantity changes. We see the quantity, but quality, we still have to look, and perhaps quality, like response to influenza would be very much dependent on, for example, cell death of alveolar macrophages in lungs and stuff like that. A lot of cell death is happening in influenza. So perhaps maybe there is a initial setup in quiescent cells. They are more predisposed to die, and that could be very interesting to study.
Wonderful. So Shandong Wang asks, sex differences in the immune responses probably affect the complex interactions among genes, hormones, and our environment. And he gives an example of nutrition on infection. Would you comment on which one of these factors, such as genes, hormones, et cetera, would be a major factor as to contribute to the sex-specific effects of the immune response?
So the environment, and what's the role of environment, Chris, in
genes, hormones, environment, kind of all of the above. What do you think is the major contributor in this case to the specific immune responses?
Well, I think the major contributor is still, like, with my mice, I know certainly very little about the epidemiological studies and the role of hormones in in humans. I just didn't have enough time to educate myself. But in mice, I wouldn't be surprised if estrogen is responsible for everything. So that ends up with estrogen, and one would wonder, like, well, so what's the big story then? But we identify genes. Estrogen is not encoded by gene, right? So it can be high and low, and the question is, how does it fit into the rest of the picture? So I think our genetic analysis provides the bottleneck, like, with these receptors, because I think, as every genetic study, it shows causation. I mean, it shows you the way things are. We need to explain them, and I guess I'm struggling with trying to explain the big picture. And so, like, my answer to that would be, at the end of this story, I believe estrogen plays a very important role.
So Sheldon Rowan asks, do you think there's an evolutionary trade-off in LPS resistance for the F1 mice? What do you think might happen if the F1 mice were in the wild and were exposed to pathogens?
Great question. I think F1 mice are great resources. I mean, everyone should study them. Well, I'm 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 joking, of course, but we feel that, well, first of all, these females, they are dropping anywhere from 12 to 16 pups. So can you imagine this in inbred? I mean, in inbred, in classical inbred mice. So they are really have heightened fertility. They are very physically different. I mean, I believe, my question to that is, I believe they have many more traits to study, and the number of genes that we identified, or the number of loci, it's a proof of that.
Wonderful. Well, unless I see any last minute questions, I'd like to thank you for your time today and for giving us a wonderful talk and stimulating my thought processes. And I'm probably going to bother you later about the genetics of immunity because I have lots more questions to follow up with you with.
All right.
Wonderful. With that, thank you everyone for attending the seminar today, and we will I believe next week we will have another seminar.
Thank you for having me.